This is Dr. Tandru Chakraborty, and today's lecture is on neurotransmitter chemistry. What are neurotransmitters? A chemical released from a nerve that diffuses to another nerve or effector cell where it causes excitation or inhibition in a specific manner. So some of the criteria of neurotransmitters are synthesis and storage in the presynaptic neuron, released by the presynaptic axon terminal, chemically and pharmacologically identical form, produces a response in the postsynaptic cell, blocked by competitive antagonists in a dose-dependent manner, an active mechanism that can deactivate or terminate the action. So these criteria neurotransmitters must have, or a chemical must have, in order to be coined the name neurotransmitters. So if you look in here, these are the different neurotransmitters. They are divided according to structure in the following way. Number one, amino acid. These are small organic molecules which has an amino group, NH2, a carboxyl group, COOH, and they are relatively small carbon backbones. So usually examples are glutamate, glycine, GABA. Amines, these are also small organic molecules. Usually they contain an amine group, NH2 or NH3+, plus, an aromatic ring, okay, very important, or an ester linkage. Examples, dopamine, acetylcholine, histamine. Peptides, these are short chain proteins stored and released from the secretory granules from the cell soma, short chains of amino acid linked by peptide bonds, can be much larger in molecular size compared to amino acids and amines. Dynorphin and enkephalins. We are not going to touch upon this. The first two are the ones that we will be focusing on. So according to function, neurotransmitters are classified according to their excitatory and inhibitory action. Before we proceed, one very important thing to remember, which we have done before, the nature of response, whether it's excitatory or inhibitory, at the postsynaptic neuron does not depend on the chemical nature of the neurotransmitter. Instead, it depends on the type of receptor being activated and the ion species that becomes more permeable. For example, we'll be doing in details how acetylcholine works. But acetylcholine, as you can see it in here, it has two functions. It can be excitatory, it can be inhibitory. For example, acetylcholine produces synaptic excitation at the neuromuscular junction, which is skeletal muscle contraction, by binding to the cholinergic nicotinic receptors, whereas the same neurotransmitter, which is acetylcholine, produces an inhibitory response, decrease in the heart rate, by interacting with cholinergic muscarinic receptors located in the cardiac muscle. Very, very important thing to understand. So, you will see in here, there are several neurotransmitters which shows both excitatory and inhibitory response, and that's based on the neurotransmitter receptors that are present on the postsynaptic neuron. So classification, acetylcholine can be both excitatory and inhibitory. Glutamate is excitatory neurotransmitter. GABA and glycine are both inhibitory. Dopamine can be excitatory and inhibitory. Non-epinephrine can be excitatory and inhibitory. Serotonin can be excitatory and inhibitory. Epinephrine can also be excitatory and non-inhibitory. So as you can see that most of the neurotransmitters, depending on the neurotransmitter receptors, they act accordingly. Let's do a primary function, how they are, what are the function. Acetylcholine is mainly responsible for muscle contraction, learning, memory, autonomic signaling. Glutamate, remember, it has only excitatory functions. It's a classical excitatory neurotransmitter. It is also related with learning and memory. GABA is an inhibitory neurotransmitter, is one of the most important inhibitory neurotransmitter in the brain. Glycine, inhibitory control of motor and sensory pathways. Dopamine is for reward and motivation. Norepinephrine, alertness, attention, stress response. Serotonin is for mood, sleep, appetite. And epinephrine is for fight and flight response. These shows major secretory sites where they are produced, and we'll go over them as we go along. So this is showing you the entire process before we proceed into the neurochemistry in details. So as you can see, there is arrival of the action potential at the presynaptic terminal. There are voltage-gated channels of calcium that are opening up. This causes activation of snare. The snare proteins are very important, which helps in the permeability of the presynaptic terminal so that these vesicles which carry the neurotransmitters can fuse. Calcium binding to synaptogen, basically this snare protein, docking and fusion of the synaptic vesicles in the presynaptic membrane. So this docking or fusion takes place. Exocytosis of the neurotransmitter into the synaptic cleft, as you can see it in here. Diffusion of the neurotransmitter across the synaptic cleft. So they move past the synaptic cleft. Binding of the neurotransmitter to the postsynaptic neuron. So they bind to the postsynaptic neuron. And once they bind, they produce a response, of course. But once their job is done, once the process has been initiated in the postsynaptic neuron, there is termination of the neurotransmitter action, reuptake it can be, enzymatic degradation, or diffusion. So going forward, for each of these neurotransmitters, we are going to learn synthesis, removal, distribution, and what are the physiological and chemical effects of these neurotransmitters. We're going to start with acetylcholine, which is also known as cholinergic neurons. The main function, as we learned, is for muscle contraction, learning and memory, and autonomic signaling. So in the brain, we are going to focus on the brain only. It is very important in the hippocampus because that's responsible for memory formation, in the cerebral cortex for thinking and attention. In the basal forebrain, it is a major source for cholinergic neurons. And in general, this cholinergic neurons or acetylcholine is very, very important for cognitive functions. So how is it made? So the synthesis of cholinergic neurons is shown in here. So we are going to follow the steps as it go along. The first step as you can see in here, glucose enters the nerve terminal by passive transport fasciculated diffusion. So by glucose, as you know, is the main molecule which is responsible for neurons to function. Glucose is the only source of fuel that neurons can take up. So glucose enters the nerve terminal by passive transport. And you can see it in here. Shown in here is a mitochondria. Glycolysis occurs in the neuronal cytoplasm, and pyruvate molecules are generated. So pyruvic acid, pyruvate is generated. The pyruvate is transported, the pyruvate here is then transported to the mitochondria, and an acetyl group derived from the pyruvic acid combines with coenzyme A present in the mitochondria to form acetyl coenzyme A, which is transported back into the cytoplasm. So acetylcholine A is made, it goes into the cytoplasm. Choline, this molecule in here, is a precursor of acetylcholine, is actively transported into the neuronal terminal from the synaptic cleft via sodium and choline transporters. And I'll show you how they are transported back. Once choline is formed, it goes back, choline goes back. This molecule comes from acetylcholine, which is in the synaptic cleft. This choline goes back into the terminal. Acetylcholine is synthesized in the cytoplasm. So acetylcholine with choline, acetyl coenzyme A with choline forms acetylcholine. Again, acetylcholine is synthesized in the cytoplasm of the nerve terminal, as you can see it in here, from choline and acetyl-CoA in the presence of an enzyme called choline acetyl transferase. Acetylcholine then is transported in the vesicle and stored, as you can see it in here. Vesicular acetylcholine transporters are there, which holds the acetylcholine in the synaptic terminal. It is then released into the synaptic cleft by exocytosis and hydrolyzed by acetylcholine esterase. So acetylcholine comes into the synaptic cleft. In the postsynaptic neuron, there are cholinergic receptors. Acetylcholine binds to it, and rest of the acetylcholine is then decayed, or you can say broken up, into choline and acetate. This choline is then recycled for the making of the acetylcholine again. So once more, glucose comes in, converted into pyruvate. Pyruvate goes into the mitochondria. In the presence of choline, which is transported back from the cleft, acetylcholine A from the mitochondria and choline interact in the presence of choline acetyl transferase and is converted into acetylcholine. These are then taken up by the transporter protein and then through exocytosis. And if you remember, this is when action potential comes in, calcium channels open, snare proteins help in the process for fusion, diffusion, and acetylcholine comes out. Once acetylcholine binds to its receptor, rest of the acetylcholine is decomposed or broken up by acetylcholine esterase into choline and acetate. Choline is recycled back to form acetylcholine. And as you can see in here, there are glial cells which can also take up acetylcholine and store it. So this actually shows you exactly what I have just said in here. to show you how choline is transported back. There are choline transporters that help in when sodium comes in, choline also moves into the synaptic cleft. This actually shows you step by step how the process takes place. So there are two major constellation of cholinergic neurons. So, so the question is, what are we learning here? We are learning the distribution of these cholinergic neuron in the brain. So cholinergic constellation, what are constellation? These are network of neuron in the brain that basically use acetylcholine as a neurotransmitter to project widely in areas involved in learning, memory, and attention, which is a main function of acetylcholine. So there are two of them. One is the basal forebrain constellation, which includes basal nuclear minors, which provides cholinergic innervation to the cortex, amygdala, hippocampus, and thalamus. The next is dorsolateral tegmentum of the pons. So if you look in here, this is the brain, this is the cerebrum, this is the diencephalon. This is the brainstem, this whole thing, which is midbrain, pons, and medulla oblongata. This is the cerebellum. So shown in purple is a basal forebrain constellation. It initiates here and then actually distributes throughout the forebrain, as you can see in here. In this cerebrum, neocortex, involves thalamus, it involves corpus callosum. And there is another constellation which is in the pons, which is in here, which actually has its projection in the different parts of the hypothalamus. It goes into the medullary, in the cerebellum also. So the next part is before we proceed, I want to actually go into one other thing is we did this, but just to be clear that how the acetylcholine is removed from the synaptic cleft. High concentration of this enzyme, which is acetylcholine esterase, is present, which actually breaks down acetylcholine into choline and acetic acid. And as such, acetylcholine does not stay into the synaptic cleft. Remember, it is essential to remember that these neurotransmitters cannot stay into the synaptic cleft for a long time. They need to be removed and recycled because if they are present at a much longer time than required, they can cause several diseases. So Alzheimer's disease has been implicated to be related, or acetylcholine has been um, is one of the major neurotransmitter which is involved in Alzheimer's disease. It is said that this acetylcholine neurons degenerate, especially in the basal forebrain. Don't forget that acetylcholine is responsible for memory and cognition. So acetylcholine synthesis decreases, acetylcholine transferase activity is reduced. So which this enzyme, if you go back, you will understand. So if you remember this enzyme, choline acetyl transferase or acetylcholine transferase, if this decreases, acetylcholine formation does not happen. And in that case, the synaptic transmission becomes impaired There is less communication, neuronal, a network weakens, and that can lead to memory loss, difficulty in learning, in new information. There's reduced attention and concentration, confusion, disorientation. These are all hallmarks, these are all symptoms of Alzheimer's disease. So, what are the symptoms? Choline esterase inhibitors. So, if there is less formation of acetylcholine, if we block acetylcholine esterase in the synaptic cleft, so whatever is made stays in that synaptic cleft. So, that helps to increase acetylcholine availability for the postsynaptic neuron, and that can improve communication of neurons, provide temporary cognitive impairment. Actually, there is no cure for Alzheimer's disease. We can slow down the process because, yeah, at the end of the day, acetylcholine has to be made in order for the neuronal transmission to occur. And since these neurons are unable to make acetylcholine, Alzheimer's disease cannot be completely, cured, but we can definitely slow down the process. So, going back one slide before we proceed, is acetylcholine, remember, requires acetyl-CoA and choline. With the presence of acetylcholine transferase, comes to the synaptic cleft, attaches to the postsynaptic neuron. Acetylcholine that is left is degraded by acetylcholine esterase. Choline is recycled. One of the major forms of treatment of Alzheimer's disease, which is related with less formation of acetylcholine, is to reduce acetylcholine esterase so that acetylcholine remains in the cleft. And also, by increasing acetylcholine transferase, acetylcholine can be increased. Alzheimer's disease is directly related with acetylcholine formation, synthesis, and release and reuptake. And in the presence of less formation of acetylcholine, it leads to Alzheimer's disease, which cannot be cured, but the process can be slowed down. Next is glutamate. So glutamate is a very important neurotransmitter because it's one of the main excitatory neurotransmitter. It is related with learning, memory, and synaptic plasticity. It occurs in high concentration in brain, glutamate, remember, okay? Although it plays a role there are many other functions because it is involved in the formation of proteins. Glutamate actually is considered to be the most excitatory amino acid because it is involved in the formation or its use as a neurotransmitter in the brain. So the synthesis, how it is made. Glutamate is made by the following steps. So glutamate is present throughout the neuron. It actually goes into the mitochondria, where it is converted into glutamate by the enzyme glutaminase. Glutaminase converts glutamine into glutamate. Glutamine is transported out of the glia into the neuronal terminal by glutamine transporters. So I want to focus at this side also. So glutamate, which is taken up from the synaptic cleft also, is converted into glutamine by glutamine synthase and is also transported in here as glutamine, which can also make glutamate. So this is astrocytes. If you remember, we did neuroglial cells, and this is also responsible for the formation of glutamine and forms glutamate. In the in the neuronal terminal, also in the neuronal terminal, this in here. So in here, the glutamate that is made, here we're talking of the nerve terminal. This entire process is happening in the nerve terminal, remember. Glutamate is packaged into by the vesicular transporter through exocytosis. It is released in the synaptic cleft and then attaches to the postsynaptic receptors. The glutamate that is not used is taken up by the neuroglial cells by glutamate transporter and then again converted into glutamine. And as I said, by glutamine synthase converts this into glutamate in the astrocytes or neuroglial cells, you can say. This glutamine is taken up by glutamine transporter and becomes the precursor molecule to make glutamate again. So here we're talking about two enzymes. One enzyme, glutamine, is converted into glutamate in the mitochondria by glutaminase. And in the astrocyte, glutamate is converted into glutamine by glutamine synthase. So two enzymes are involved in this process. And remember, removal of these neurotransmitters from the cleft is glutamine transporters are the main ones that help in the recycling of glutamate from the in this region. Glutamate is taken by high affinity sodium dependent reuptake mechanism into the nerve terminal, and glial cells play a very important role in their reuptake process. This shows you the steps in which it synthesized and the recycling of the glutamate-glutamine cycle. So the distribution, this is present in cerebral cortex, hippocampus, cerebellum, thalamus, brainstem, spinal cord. Pretty much it is present everywhere in the brain. These are the major location and its main role. It has several roles, the main role being in the cognition and perception, memory and learning. Glutamate is involved. Physiological and clinical consideration of glutamate. Some of the important physiological and clinical consideration relevant to glutamate are the following. The first one is glutamate has been implicated as a transmitter in a variety of circuits in the brain. For example, excitatory amino acids may be involved in learning and memory processes, as well as several motor functions. Glutamate has also been implicated in some chronic neurological condition, such as shown in here in the amyotrophic lateral sclerosis, also known as Lou Gehrig's disease. It's a progressive neurodegenerative disorder that primarily, mainly affects motor neurons. So, if you remember, motor neurons are the ones that affect the skeletal muscles primarily, and this disease leads to muscle weakness and paralysis. So, what it is characterized by the degeneration of motor neurons, as we just said, and it is important that the person suffering from this disease exhibit wide range of muscle, dysfunction, atrophy, weakness. The mechanism has been said to be because of glutamate cytotoxicity. What it is, let's learn about that a little bit. Prolonged stimulation of neurons by glutamate, which is an excitatory amino acid, results in neuronal death or injury. This process, whenever there is overstimulation of excitatory neurons, it leads to excitotoxicity, which is toxicity related to excitation, in which loss of postsynaptic neuron predominates. So the presynaptic neurons become so excited, it has become so excited that the firing becomes so continuous that it can lead to cerebrovascular stroke. Okay, so it is important to understand that excess glutamate that accumulates in the synapses causes overstimulation of the neurons, causing excessive calcium entry, leading to neuronal damage. The drug involved is rirosol, and we're going to learn about this entire process in the next slide. So this side shown in green is the normal nerve excitation. Neuron, how it diffuses out, generates, basically excites the spinal cord, which has its action through the motor neurons into the skeletal muscles. In this, you can see this is a normal neuronal cell, which basically is activating the muscle cell. In case of amyotrophic lateral sclerosis, when there is deterioration of the motor neurons, these motor neurons that are coming out from the spinal cord, they do not function properly. As a result, when they do not function properly, there is an impaired signal transmission. This impaired transmission leads to less neurotransmitter to be released if the glutamate muscles do not receive adequate stimulation, and as a result, which can lead to ALS affected neurons in the motor. So overstimulation of glutamate causes this, the synaptic transmission to be damaged. And this, the neurotransmitter, as a result, cannot act on it, as a result, which leads to dysfunctioning of the muscle stimulation. So the next that we need to understand is a very, very important neurotransmitter, which is called GABA. Okay. I want to emphasize here one very important thing is GABA is a inhibitory neurotransmitter. So when I say inhibitory neurotransmitter is basically it is responsible for reducing the activation. So a balance is needed with regard to excitatory and inhibitory neurotransmitter. So GABA primarily acts as an inhibitory neurotransmitter in the brain. It is in the motor control, it works in the basal ganglia. So if where is the basal ganglia, let's learn about. So, a basal ganglia are a group of subcortical brain nuclei that are found in the forebrain. So, if you remember when we learned brain, we learned the forebrain part, and GABA is mainly responsible for voluntary motor activities, related with habit formation, reward processing, and emotions. So it has many functions. Some of the key functions is again motor control. It acts as a gatekeeper, choosing which motor actions to be effective, which needs to be inhibited. It is related with habit and reward, that they play a major role in habit formation, like addiction, motivation, and emotional processing. And it is also, since it is involved in the basal nuclei in the forebrain, it is involved in executive decision making and memory formation also. Okay, so again, GABA is an inhibitory neurotransmitter. It affects the basal ganglion neuron in the basal ganglia. It balances the excitatory signals, and of several neurons, and dopamine is one of them, as written in here. So, uh, one important thing to understand in the next slide we'll go and is this actually shows you how the entire process of synthesis and release of GABA takes place. So let's go over it from here. So glutamate is converted into glutamate, okay, in the mitochondria. And GABA is formed by, this glutamate acid decarboxylase. This enzyme decarboxylation takes place. This is known as GAD, glutamate acid decarboxylase, GAD it's called. We usually call it GAD, which is present almost exclusively only in the GABAergic neurons, means only the neurons that produce GABA, it is present in GABAergic neurons. GABAergic neurons. GAD is not present in neurons using glutamate as a transmitter or in glia. Very important to understand. So GAD is present only in GABAergic neurons, and that is the reason glutamate is converted into GABA. And once converted, synthesized, it's taken up by the vesicles. And one important factor in here is to remember that the formation of GABA is related to, it requires a coenzyme. Okay, what is the coenzyme? Vitamin B6 is required in this formation. Very important to remember this. It is once it utilizes the GAD and converts it into GABA in the presence of vitamin B6. It is vascularized and then, of course, released in the synaptic cleft. GABA is then taken up by the glial cell. In glia, GABA is converted into glutamate by the mitochondria, as you can see it in here, by an enzyme which is known as glutamate synthase. Glutamate is converted into glutamine by the enzyme glutamine synthase, and glutamine then subsequently again through the glutamate transporter goes back to form glutamine. One other thing to remember here is the conversion of GABA into glutamate is done by another enzyme, which is known as GABA transferase, also known as GABA T, which converts GABA into glutamate, and then the enzyme glutamine synthase converts into glutamine. So distribution of GABA is throughout And we'll do that. One, I just want to make sure that you have understood the synthesis of GABA, that it is important that glutamate is taken up by the presynaptic neuron, largely supplied by the surrounding neuron. So glutamate is the precursor molecule, and then it converts into GABA, then released through calcium-dependent exocytosis like any other processes. And then in the postsynaptic neuron, there are GABA A and GABA B receptors that have influence on the neuron and produce an inhibitory effect. This is important to remember, that the GABA receptors on the postsynaptic neuron are inhibitory. So they inhibit the action of other excitatory neurotransmitter. So going forward, distribution, GABA is found in high concentration in brain and spinal cord, and is absent in peripheral nerves and tissues, which means in the spinal nerves it's not really present. Some of the physiological and clinical effects of GABA is, number one is GABA, as I said, is an inhibitory transmitter in many brain circuits. For example, GABA is the primary inhibitory neurotransmitter in the Purkinje cells in the cerebellum. If you remember cerebellum, those white structure that throughout the cerebellum are the Purkinje cells, and it acts as an inhibitory neurotransmitter in there. Alteration of GABAergic circuits has been implicated in several psychiatric diseases like Parkinson's disease, Huntington disease, and Alzheimer's disease. So here we are actually talking about Parkinson's disease. So, Parkinson's disease is a chronic progressive neurological disorder, and it involves mainly it is related with movement, motor control. Okay, so excessive inhibition of motor pathway. That means when GABA is inhibited a lot, or if the release of GABA is not enough, that causes reduced stimulation of the motor cortex, and the motor movements is inhibited. So if you look in here, if GABA, this is a normal dopamine being secreted in the substantia nigra neurons, and when GABA is produced in less amount, what happens is these are it is depleted, and when this is depleted, you can see this is the normal substantia nigra and this is depleted substantia nigra. As a result, the motor neurons are affected. And the reason, there are many reasons. One of them can be if your diet contains less vitamin B6. Remember, vitamin B6 is a coenzyme that is required for dopamine formation. I'm sorry, for GABA formation. If you remember, a dietary deficiency of vitamin B6 can reduce GABA synthesis. So vitamin B6 is required for GABA formation. So in its absence or in its production in less amount, it can lead to less production of GABA synthase, which is required for GABA formation. And there are several treatments to help with Parkinson's. We really don't have a medicine that can help in the process, but we can delay the progression of Parkinson's. There are certain treatments like deep brain stimulation. Certain drugs can indirectly help with GABA signaling. So if you remember the last slide, we showed you Parkinson's when GABA is produced in a less amount. There is also another disease, which is epilepsy, which is seizure. When epileptic seizure can be facilitated by lack of neuronal inhibition. That means increase in the inhibitory transmission. So if GABA is present in large amounts, what will happen is it will start to um, inhibit the neurons. There are certain disease, the certain medicines available that are useful because it inhibits GABA transaminase. So if you go back a slide, trans. So if you remember, we did GABA transaminase is present, which actually converts into glutamate. This medicine, which is known as Velproc, actually Valproic, Volproic, inhibits GABA transferase. So as a result, GABA remains, you can understand if this is not converted into glutamate, GABA actually remains in this region, which helps a lot in the process. There are barbiturates. They act as agonist on the postsynaptic GABA receptors and are used to treat epilepsy. So sometimes GABA is required in order so that inhibition can take place. And if GABA is present in high amounts, sometimes it can lead to several psychotic diseases as well. We're learning about GABAergic neurons, physiological and clinical implications of GABA. First one is Parkinson's disease. It is, Parkinson's disease has been implicated to be mainly because of dopamine deficiency, which we will be doing. And dopamine is considered to be the hallmark of Parkinson's disease, but we have found recent research has shown that GABA, GABA has been shown to be implicated in Parkinson's disease as well, and is considered to be a critical regulator of both motor and non-motor symptoms associated with Parkinson's disease. So, as we have learned that GABA is an inhibitory neurotransmitter. So, GABA role in the dysfunctioning of Parkinson's is mainly because it helps in regulation of excitatory and inhibitory signals. And when GABA does not function properly, um, and uh, and can cause excessive inhibition that can lead to several diseases. And in the presence where GABA does not function properly and causes less inhibition, that can also lead to several diseases. So there is a theory called the GABA collapse theory. It's an hypothesis mainly, and it is considered like a decline in GABAergic inhibition in the basal ganglia leads to overstimulation of of the signals. As a result, it can lead to rigidity, bradycardia, posture instability, some of the many signs of Parkinson's disease. So, Parkinson's disease is a chronic progressive neurological disorder that mainly affects movement. Um, this causes excessive inhibition in the motor pathways, reduced stimulation in motor cortex, and difficulty in initiating movement. So these are some of the side effects of Parkinson's, which may be related to GABAergic neurons. And why the real cause of Parkinson's is really not known, but some of the reasons are, if you remember, formation of GABA requires vitamin B6, and it has been shown that if the dietary there is a reduced B6 or dietary deficiency of vitamin B6 can cause reduced formation of GABA synthase, which can cause a reduction in formation of or less formation or loss synthesis of GABA, which can lead to Parkinson's disease. Some Parkinson's disease treatments, is not well defined, or we really don't have a proper treatment strategy or medicines related to it. But there can be deep brain stimulation that alters GABA circuits. Certain drugs can indirectly help with GABA signaling. The second is epilepsy. It's a chronic neurological disorder in which the person has recurrent unprovoked seizure caused by abnormal electrical activity in the brain. So you can see it in the figure. This is the normal brain, and when there is continuous stimulation going on by excitatory neurotransmitters, it can lead to seizure. So if you remember, GABA is an inhibitory neurotransmitter, and if present in very low amount, this inhibition does not take place. As a result, there is uncontrolled neuronal firing that can lead to seizure. Low GABA function is mainly results in convulsions, loss of consciousness, and muscle spasm. There are some treatments available. Some most anti-epileptic drug enhance GABA action. So if GABA action is increased, inhibition is increased. As a result, the firing reduces. Increasing GABA levels strengthens GABA receptors, reducing GABA breakdown. This helps to calm the brain. The firing reduces. Catecholamines, interesting. This is a group of three neurotransmitter. They are primarily an excitatory neurotransmitters, and there are regions where in your body where it can act as an inhibitory neurotransmitter as well. But primarily, these are excitatory neurotransmitter in the brain. One important thing to remember here uh, is for catecholamines, the starting precursor molecule or the starting molecule is tyrosine amino acid, and tyrosine is a precursor of three amine neurotransmitter, which is these are the three: dopamine, norepinephrine, and epinephrine. The neurons that produce dopamine is known as dopaminergic neurons. Norepinephrine, Ne, is called noradrenergic neuron, and epinephrine is known as adrenergic neuron. So, and there's a specific reason why this naming is done, and as we go along, you will understand it better. So this is actually shows you what is the biosynthesis of catecholamines, how they are made. So the starting material is tyrosine, and for tyrosine, the starting material phenylalanine. We will consider tyrosine as a starting material for all the three catecholamines. Tyrosine, in the presence of tyrosine hydroxylase, which is a rate limiting step, is converted into dopa, which is converted into amino acid decarboxylase, which is decarboxylation takes place where dopa is converted into dopamine. So this is an important enzyme that is present in some neuron, which converts dopa into dopamine. Okay, and then dopamine, in the presence of dopamine beta hydroxylase, which is hydroxylation process, converts dopamine into norepinephrine, and norepinephrine in the presence of phenylethanamine N-methyltransferase, and we will call it PNMT, is converted into epinephrine. The question is, does one neuron, when it has all of these enzymes, why it stops in formation of dopa and not completely convert tyrosine into epinephrine? And you can see that it's shown in here that part of it occurs in the cytoplasm, part of it happens in the storage vesicles, and then again part of it happens in the cytoplasm. We'll learn about this in the next slide, but let's do this again. Tyrosine, which is a precursor molecule, as you can see, in the presence of tyrosine hydroxylase, it actually loses its hydroxylation, so it becomes dopa. And in the presence of amino acid decarboxylase, it further becomes dopamine. Okay, dopamine, and then in the presence of dopamine hydroxylase, converts into norepinephrine, and then PNMT converts it into epinephrine. So how does it happen? We are talking about everything happening in the neuronal cell. So tyrosine is transported into the axonal end, you can consider, and tyrosine in the presence of tyrosine hydroxylase. Everything is, the abbreviations are given here. Tyrosine hydrolase is converted into dopa. Dopa in the presence of dopa decarboxylase is converted into dopamine. This entire process is happening in the cytosol of the neuron. Okay, now it is in this region, you just have to understand this is happening in the axonal terminal. At this point, remember, the neurons that have dopa decarboxylase produces dopamine, and they are responsible, they are known as dopaminergic neurons, right? And this dopa can then actually be go into vesicles and then be transported. But some of the neurons, apart from having dopa decarboxylase, also has another enzyme which is known as dopamine beta hydroxylase. And what it does, it converts dopa into Ne, which is norepinephrine. Okay, this is happening in the remember, in the vesicle. In the vesicle, norepinephrine is present. Now, in some neurons, this is only present, DBH is only present, which converts dopamine into norepinephrine, and norepinephrine becomes the neurotransmitter for that neuron. Now, in some neurons, in addition to norepinephrine DPH, there is another enzyme present, which we just PMNT, which is this PNMT is present in there, and which actually is present in the vesicles again. So instead of norepinephrine, what it does, it goes into the cytosol again, and this cytosol, in the cytosol, when it is present, when PMMT is present in the cytosol, it converts any into epinephrine. Now, this epinephrine that is present in here in the presence of PMMT has been converted into epinephrine, now can be packaged back into the synaptic vesicle and now can be used as a neurotransmitter. So here we are, learning about three neurotransmitters being formed from tyrosine. So one is dopa, or from L-dopa or dopa, which is converts into dopamine. Everything is happening in the cytosol. Then it goes into synaptic vesicles, where it has this enzyme, which is very important, which is dopa decarboxyl. This was dopa decarboxyl, and this is dopamine beta hydroxylase, the dopamine DPH, beta beta hydroxylase. Hydroxylation happens, which is dopamine beta hydroxylase. Which converts dopamine into Na. And when Na is formed, now these vesicles are ready to transfer this norepinephrine as a neurotransmitter. When this comes into the cytosol, now there are certain neurons that have another enzyme, which is PNMT, which converts norepinephrine into epinephrine. And this is happening in the cytosol. Once this is happening in the cytosol, one epinephrine is formed. Again, packaging is required, so it gets packaged into the synaptic vesicle, and then those neurons are now considered to be epinephrinic or adrenergic neurons and can have their own function. So next, we are going into, we are learning what are the synthesis of dopamine, and we did this a little bit. We are going to do it in a more pictorial manner. So the amino acid tyrosine, is converted. Amino acid tyrosine it's present in all food products and can be synthesized from phenylalanine. We learned that, right? But we say that the starting point of all of these three neurotransmitters is tyrosine. In the cytoplasm of the dopaminergic neuron. Now we are using the word dopaminergic neurons. You remember, because we are learning about dopamine only. The tyrosine is converted into dihydroxyphenylalanine dopa by the tyrosine hydrolase. So if you look in here, by tyrosine hydrolase, L-dopa is formed. L-dopa is converted into dopamine by dopa decarboxylase, which we did, okay? Now, dopamine is then actively transported into the storage vesicles by vesicular transport. We know that it gets packaged into vesicular transport and then is released out. This is an active process. In the dopaminergic neuron, dopamine remains unchanged in the storage vesicle and of course is released by the process of exocytosis. Dopamine released in the synaptic cleft is actively transported back into the neuronal transport. So this again, if you remember, dopamine that is present gets attached to its receptors, and the reuptake is taking place. Now we're talking about the reuptake. This reuptake process is very important, by which dopamine and other catecholamines are removed from the synaptic cleft. Some of the crucial process by which it takes up is of course dopamine that is attached to its receptors and does its action, but the remaining dopamine then enters into the neuronal terminal, is destroyed by the mitochondrial enzyme monoamine oxidase. MAO. Okay. Some of the dopamine that is released in the synaptic cleft, about 10% of that, is actively transported into the effector cells. And effector cells means it can be liver. The process is called reuptake 2, and dopamine is, and once it enters the effector cell, and primarily by the enzyme catechol O methyl transferase, which is COMT, catechol O methyl transferase. Uh, it is again both of them actually is responsible for reuptake and for giving it back to the axonal terminal. Uh, so this reuptake by the liver is important process, and some are some actually go back to the axonal terminal, and dopamine is recycled back and used again. Some dopamine in the liver by MAO and COMT can be destroyed. The end products of metabolism um, are actually released through urine. Let's go over this again once more. Dopamine's main function is emotion, motivation, thinking, and perception. The starting material, we'll consider it as tyrosine. Tyrosine converts into L-DOPA by tyrosine hydroxylase, which is the rate limiting step. And L-DOPA is then converted into dopamine by DOPA decarboxylase. Dopamine then actually is vascularized, in the vesicular transporter is used, and through exocytosis comes out. Dopamine in the synaptic cleft can be converted into with the help of certain enzymes, and and recycled back into the synaptic cleft by transporter proteins, or it can be actually even be destroyed by some effector cells in the liver, like liver, by MAO and COMT, and the products can be excreted out as in the urine. Distribution of dopamine is huge. In the brain, dopamine has four pathways by which it actually has its action. Number one is a necrostriatal pathway. So necrostriatal pathway shown in blue is in here. Its origin is in the substantia nigra, and actually then it passes into the striatum. Okay. Its main function is it controls voluntary movement and muscle coordination, and it actually helps to smoothen muscle movement. Then comes the next one, which is a mesolimbic pathway, which is very important, shown in dark blue, this in here. Its origin is in the ventral tegmental area, as you can see it in here. Ventral VTA, it's called ventral tegmental area in the limbic system. It is related with reward, motivation, pleasure, emotion. So it's very, very important pathway. And the next comes is mesocortical pathway, shown in red, which actually covers a lot of area. Again, it starts with around the VTA and goes in the prefrontal cortex area. As you can see, the cortex in the cerebrum is affected. And its main function is thinking, attention, decision making, because it's in the cortex area. Then the last one is tuberoinfundibulum pathway, and It is a hormonal pathway control. It starts from the hypothalamus, like it shows you here, it's in the pituitary, and then actually it goes into the hypothalamus and has its action, and it is responsible for, it helps in the hormone production. So all of this is written in here, how the negropatwell pathway is in the movement. It starts from substantia nigra and projects to the stratum. What is its function is voluntary movement, coordinates muscle activity, and its damage can cause Parkinson's disease. Mesolimbic pathway, very important, starts with the VTA, which is ventral tegmental area, and then projects to the nucleus albicans, amygdala, and hippocampus, which is the part of the limbic system. It is related with reward, motivation, pleasure, and addiction. Overactivity can lead to psychosis and addiction. Mesocortical pathway is related with cognition and mood. Again, starts with PTA and projects to the prefrontal cortex. It's related with decision making, thinking, attention, emotional control, etc. And its dysfunction can lead to depression and schizophrenia. Tuberoinfundibular pathway is related with hormonal control. Origin, you can consider hypothalamus or pituitary. It projects from pituitary to hypothalamus or hypothalamus to pituitary, either way. Uh, And it regulates hormone release. And in its absence or in its malfunctioning, there is hormonal disbalance. So some of the physiological and clinical implication um, is when we were talking about GABA neurons or GABAergic neurons, if you remember, we were talking a lot about dopamine, and dopamine is the primary neurotransmitter involved in the Parkinson's disease. And again, Parkinson's disease is a chronic progressive neurological disorder and mainly affects movement, motor movement. It occurs in brain cells that produce dopamine. Um, and generally, the dopaminergic neurons do not produce dopamine in high amounts. Since the levels fall, and the GABA activity becomes abnormally high in motor circuits. So dopaminergic neurons decrease, and GABAergic neurons activity increase. As a result, the motor functions, the motor movement become uncontrolled. Okay. Um, there are other psychotic disorders like schizophrenia, which are also implicated with related with dopaminergic neurons. Dopamine actually uh, is uh, competes with cocaine, cocaine drug users. Dopaminergic neurons and cocaine users or cocaine they compete with each other. Dopamine is part of the reward system, as we just learned, and it produces a feeling of pleasure and motivation. So it is related to feel motivated. Uh, what cocaine does actually, it blocks dopamine reuptake, preventing it from removing from the synapses. So dopamine remains in the synapse. As a result, what happens is there is a feeling of intense pleasure, which we call high or euphoria, and increased energy. Repeated use can lead to addiction. I'm going to go over the next slide first. Uh, no. Um, so repeated use can cause addiction, and long-term effects is that remember, using cocaine, it can be detrimental, can have detrimental effect. Long-term effect is dopaminergic system gets damaged. Um, as a result, natural pleasure that you will get from different type of actions or episodes in your life decreases, and it can lead to depression and addiction, that is craving of more and more cocaine. And the the treatment, of course, is it is to stop the use of cocaine. Treatment focuses on behavioral and psychological therapy. Okay. Next comes norepinephrine. If you remember, this is also known as noradrenergic neurons. And the starting material for this is the same. Um, is tyrosine, the starting material, and conversion of tyrosine to dopa is tyrosine hydroxylase, which is a rate limiting step, and then it becomes dopa, okay, for dopa decarboxylase. Now, dopa, in dopaminergic neurons, this is the final step, and it is released out. But in norepinephrine, this dopa actually gets packaged into the vesicle. Okay, when it gets packaged into the vesicle, dopamine beta hydroxylase converts it into norepinephrine, and norepinephrine then is in the vesicular form, then is excreted out by exocytosis and comes into the synaptic cleft. Again, the same pathway, norepinephrine can be taken up by its transporter back into the axonal terminal for reuse again, and or it can be destroyed by liver, like MAO and COMT can act on it, and the remnants of it can be excreted out through urine. So, norepinephrine is related with wakefulness, alertness. It improves attention and focus, enhances learning and memory, regulates mood and motivation, supports stress adaptation. Norepinephrine has, it's not one function, it has several functions. And remember, norepinephrine is pretty much distributed, everywhere. It is primarily present in locus coeruleus, located in the pons, but the function is there is various functions of norepinephrine. So it has its immediate effects on the cerebral cortex, in the hippocampus, hypothalamus, and cerebellum. So this pretty much covers the entire brain. Okay. Um, so this actually, the physiological and clinical effects of it, at normal levels, it is mainly used for alertness, focus, stress response. Very, very stress epinephrine, norepinephrine system is flight or flight response, and normal level, it helps with stress. When it is present in low amounts, it causes fatigue, poor mood, which can, if prolonged, can cause depression, attention problems. And again, if it is present in high amounts, then it can cause anxiety and high blood pressure, which can lead to panic attacks, hypertension, and insomnia, which is lack of sleep. Epinephrine is a neurotransmitter and is an hormone also, and it is best known to be a stress hormone, and has its implication in fight or flight. So we did this before. Again, the starting material is tyrosine, converts into L-dopa, which converts into dopamine. Then it goes into the vesicles, where it is converted into norepinephrine. After it is converted into norepinephrine, norepinephrine comes out into the cytosol, and in the presence of PMNT, it is converted into epinephrine. Epinephrine then vascularized goes into vesicles, and then exocytosis takes place, and epinephrine comes into the synaptic cleft. This in turn, after it attaches to its presynaptic receptors, I mean postsynaptic receptors, then it can, rest of the epinephrine can be recycled by the transporters, or it can be degraded by liver. There are several other neurotransmitters, and we are going to touch upon one, which is echinocannabinoids. This is how the cannabinoids uh, work, cannabinoids function. So let's go over that a little bit. It's very interesting. So, what are endocannabinoids? Endocannabinoids are natural chemical messengers produced by neurons in the brain. Okay, there are two main endocannabinoids. One is anandamide, and the other is 2-arachidyl glycerol. So these how they endometers, and they are not stored in vesicles. They are made only when they are needed. Okay, so synthesis of endocannabinoids. They, as you can see, this is the presynaptic neuron. This is a postsynaptic neuron. And in the postsynaptic neuron, when the signals is very strong, there is a huge amount of calcium levels increase. When the calcium levels increase in there, um, and with high calcium, it activates the enzyme that convert membrane lipids into endocannabinoids. So these when the levels of calcium in the postsynaptic neuron increase drastically, they call on the enzyme to produce endocannabinoids. And when endocannabinoids are made, um, they are not stored, they are made as needed, and then they actually act on the presynaptic neuron, as you can see in here. Endocannabinoids basically travel in the opposite direction, so that is from postsynaptic neuron to the presynaptic neuron. And then it's called retrograde action, actually. So by basically by diffusion and through the help of CB1 receptor, they bind to this receptor in the presynaptic terminal, and they are usually G protein coupled receptors. Um, and they inhibit the calcium channels in here. And when these calcium channels are reduced, what it means is fewer vesicles containing neurotransmitters will fuse. Less neurotransmitters are going to be released, like glutamate or GABA. And so basically, it acts like a brake for neurotransmitter action. So it's not a very common phenomena, but it does have an effect in slowing down the signaling process. So how they are terminated, they can be terminated by several enzymes. And some of the physiological importance of it is it's a retrograde signaling, as I said. As you can see, it's from postsynaptic neuron to the presynaptic neuron. They regulate synaptic strength, prevent overexcitation, which can happen. They actually can control pain's perception. Sometimes tremendous upon pain happens, and cannabis is given, and it's related with pain. They regulate appetite, mood, stress, and to a certain extent, they can support learning and memory. It acts as a feedback control system in the brain. Okay, so this is one other example of other neurotransmitters. Really doesn't work like a neurotransmitter, but acts as a regulator for neurotransmitters. So here, I've given all the steps in which it works. So this ends the chapter. Before we do that, I would like to actually go over the slide that we had started with, pretty much with all the neurotransmitter and its action, its function. So we did acetylcholine. It acts mainly as a excitatory, and it can act excitatory and inhibitory in the CNS, related with learning memory. And acetylcholine receptors are present in several places in the basal foramen, brainstem, and it's secreted and has its influence. Glutamate is one of the primary excitatory neurotransmitter. It's mainly responsible for learning and memory. GABA is a very important inhibitory neurotransmitter in the brain. Dopamine can be both excitatory and inhibitory, but primarily excitatory. It is related with reward motivation, and it's the secretory places are substantia nigra and BTA. Norepinephrine, again, is primarily excitatory, but there are places of inhibition, but it is related with alertness, stress response. Let's do epinephrine first. It is also an excitatory, but at times it has inhibitory action. It is mainly for flight or fight response. Serotonin is an excitatory. At times it can be inhibitory also, related with mood, sleep, and appetite. So the apart from the naming and whether its action is excitatory or inhibitory, we did how it is synthesized, how it's removed, what are the distribution, and what are the physiological and clinical implication in this chapter. So this sums up the neurochemistry of of neurotransmitters. Thank you.